ASN grad. Welcome to the ASN Graduate Research Award Symposium. I want to thank you all for the attendance. Special thanks to the speaker for making time on a Friday afternoon to give their talks. The American Society of Naturalists is hoping to establish a new tradition, a special symposium at the virtual evolution meeting, spotlighting our past student research award winners. And the goal is to celebrate and amplify the work of these award winners. So we are so excited to hear about the diverse careers that our past award winners pursued. We currently have 6 speakers. For the record, each speaker will have about 12 minutes for the talk and 3 minutes for questions, ideally. Our first speaker is going to be Takuji Usui. He's a postdoctoral fellow at the University of Toronto. He explores how ecological and evolutionary processes interact to shape changes to species distributions and community structure in an era of global change. Unfortunately, he's not here with us because he's on a plane right now. But he provided the record of his talk, which I'm going to play in a few seconds. Can you all see my screen?
Yes.
All right, so we're gonna stick to the time. Exactly at 5:30, I will play it. Make sure you all ask questions using the Q&A box. Specifically for Takuji, he's going to provide his email by the end of his talk in case you want to reach out. All right.
Hey, hi everyone. Thank you so much for coming to my talk, and my sincere apologies for not being able to join in with you all live, as I will sadly be on a plane when this is happening. So my name is Takuji. I am currently a postdoctoral fellow at the University of Toronto. And yeah, today I'm excited to talk about a project that I pitched during my PhD with Amy Angert at the University of British Columbia, which was made possible by the generous funding from the ASN Student Research Award. So I am a plant evolutionary ecologist, and my research program explores how ecological and evolutionary processes interact to shape species distributions and patterns of biodiversity across space. And this is, of course, a fundamental question to many of us in evolutionary ecology, but also is an urgent question to address, given that species distributions are rapidly shifting with climate change as species try to track suitable climatic conditions. And one critical question as climate change rapidly unfolds is whether we will see widespread range contractions as populations at the warm range edge, such as at the lower elevation or lower latitudes, can no longer persist under future and warmer climates as they're exposed to thermal environments near or beyond their upper thermal limit, or whether these populations will be able to persist demographically through rapid adaptive evolution to warming, which may rescue these populations from local extinction and range contraction. But an important consideration to this question is that at the same time as rapid warming, range edge populations are likely also experiencing intensifying interspecific competition. For example, as more warm adapted species move their distribution higher up in elevation or latitude and compete with cooler adapted resident species. And so given that demographic and evolutionary responses to warming may also be shaped by community context, complex but important challenge is to understand whether local extinctions and range contractions will be more severe with this dual stress of warming and competition, or if populations at the range edge will be able to adapt fast enough to warming, even in the presence of interspecific competitors. So in thinking about how competitors will alter adaptation to warming, I think we conventionally assume that the presence of a competitor will constrain adaptive evolution, and this could occur through a demographic pathway where competitors lower the effective population size of the focal population, and intuitively this also lowers the amount of genetic variation that can then respond to selection. But somewhat counterintuitively, though, an alternative and interesting prediction from theory suggests that competitors could also promote thermal adaptation, and this could occur, at least in theory, if there is a shared or a positively associated trait response to both competitors and abiotic stress. So let's say, for example, selection for longer roots makes you a better competitor, but also allows you to uptake resources more efficiently under heat stress. And in such cases, the presence of competitors could provide an additional selective push towards the changing environmental optimum, and therefore may promote adaptation and importantly, buffer populations from range contractions. So to test these contrasting and competing hypotheses on whether interspecific competitors constrains or promotes adaptation to warming, I used a spatial experimental evolution approach using duckweeds as a model plant community, and with funding from the ASN Research Award, constructed these replicate miniaturized ranges within which I could experimentally impose climate warming and track eco-evolutionary processes naturally unfold across replicate communities. So duckweeds are aquatic freshwater angiosperms, which are extremely amenable to experimental evolution work, where these plants propagate clonally during experiments and have a rapid generation time of 2 to 5 days, and where evolution can be quantified easily in experimental populations by tracking changes in the relative frequency of different genotypes. And because several species of duckweeds co-occur with each other in nature, they are also useful model systems to understand how evolution unfolds in competitive plant communities. So using duckweeds in our experimental landscapes, in our experiment, we first imposed an underlying temperature gradient across space from benign temperatures at the range core to hotter and more stressful temperatures at the range edge. And we started our experiment by initially placing 11 unique genotypes of our focal lemna species, placing each genotype evenly across space to ensure equal genetic diversity. And then in half of our landscapes, we also introduced a single genotype of our more warm-adapted Spirodella competitor species. We then let the communities naturally grow and populations evolve across space and over time, allowing for local dispersal and growth. And once we let the landscapes fill out, we then simulated climate change by shifting this underlying temperature gradient across space, such that the range edge is now experiencing greater temperature stress. We then let the communities naturally grow and evolve again, and in total, we experimentally evolved the populations for over 1 month, or around 15 generations. And after evolution, we then conducted a fully reciprocal transplant of our focal lemna populations between the range core and the range edge, as well as between sites with and without competitors. And by comparing the population growth rates of our transplanted populations, we could then test if populations were more or less adapted to local climatic conditions at each site, if they had evolved with or without competitors. So getting back to our question on how competitors alter adaptation to warming temperatures at the range edge, on our x-axis here, we have our measure of local adaptation as measured by home advantage, which is the growth advantage that populations that evolved at that particular site have over other transplant populations. On our y-axis, we just have our posterior densities from our Bayesian models. So when we first look at populations that evolved alone without interspecific competitors, we see that there is no local adaptation to warming at the range edge, with the mean home advantage of range edge populations falling close to 0. To our surprise, though, when we look at range edge populations that had evolved with a competing species, we see that adaptation to warming is possible. And in fact, range edge adaptation is only possible when populations had also evolved with an interspecific competitor. So we can dig a little bit deeper into what's happening here and first confirm that these evolutionary outcomes are also underlain by evolved differences in ecologically relevant traits. And so to do that, we looked at evolution in a number of traits relevant to thermal performance. And two traits that could be particularly important to warming are CT max, the maximum temperature beyond which growth is no longer possible, and thermal performance breadth, which is the temperature range at which near-optimal growth is maintained. And for populations from the range core in the blue here and the range edge in the red, we can estimate the evolved or genotype-weighted trait values after evolution. And when we do this, we see this pattern where there are not strong differences in CT max or thermal breadth between range core and range edge populations when populations evolve without a competitor. But where populations evolve in the presence of interspecific competitors, again, we see this pattern where range edge populations are able to evolve significantly higher heat tolerance in both CT max and thermal breadth compared to range core populations. So we can confirm that adaptation at the range edge is likely driven by the evolution of these thermal traits, but still this begs the question on how this could actually be happening and whether, as predicted by theory, competitors could be promoting adaptation because the evolutionary response to competition and warming is aligned in some way, such that responding to selection from competitors also helps to respond to heat stress. So to directly test this theory, we also estimated evolution in a range of traits thought to underlie competitive ability, including propagule propensity highlighted here, which we assumed that a higher propensity to produce more offspring plants would be more associated with greater competitive ability. And when we look at the evolution of this competitive trait, we find that propensity or competitive ability generally increases at the warming range edge compared to the range core, but again, more strongly so in the presence of competitors. But importantly, when we look at the association between the evolution of competitive ability and the evolution of CT max or thermal breadth, in line with theoretical expectations, we find that these traits are positively associated, where populations that evolve greater heat tolerance also evolve greater competitive ability. And so overall, these results are in line then with a potentially overlooked mechanism where if traits responding to competition also directly increases fitness under warming or are positively associated with traits conferring thermal tolerance, then competitors are able to facilitate rapid adaptation to warming, counter to what is commonly assumed. So to conclude here, given that competitions are enabling adaptation to warming, and that this seems to be done as competitors provide this selective push towards evolving heat tolerance, does this finally mean that competitors can demographically buffer range edge populations from contractions? So an advantage of doing this kind of experimental evolution work is that we can also measure the magnitude of range contractions directly from our experimental landscapes. And when we do this as predicted, we observed that range contractions to be more severe in landscapes without competitors, while the magnitude of range contractions seem to be significantly buffered in the presence of competitors. And so I think this really highlights the need to account for the feedback between community context and evolution when predicting range contractions, and that doing so can reveal key mechanisms through which interactions, species interactions can alter persistence at the warming range edge. And so in my research program, I have been continuing to use various experimental evolution approaches to test key questions on the spatial organization and reorganization of populations and communities, including on how we can better predict and understand the eco-evolutionary dynamics of range-expanding populations and communities, and on how competing species, such as in our experimental landscapes, rapidly evolve mechanisms to coexist and assemble over space and time. And in my postdoctoral work with Megan Fredrickson at the University of Toronto, I've also been expanding beyond competitive interactions to explore how positive interactions, and in particular, how co-evolution between duckweed plants and their microbial symbionts influence species range dynamics. And more recently, I've also been working with Stephen Wright, integrating genomic and epigenomic approaches to understand the demographic and the evolutionary origins of the common duckweed and why these plants are so commonly found all over the world. And lastly, we've also been really expanding the empirical resolution and throughput of our duckweed experimental system by collaborating with both software and mechanical engineers. And so I think we are now at a really exciting stage where we can start to think phenotypic variation at the individual plant level and how this influences evolution, population, and community outcomes across space and over time. And I will end by saying that this duckweed system, which we managed to start during my PhD, is in large part due to funding from this student research award, continues to live on and has now spurred on many more projects with many more students. And I will say a huge thank you to that for ASN for making science happen and also for organizing this symposium. And with that, I'll be happy to take questions through the contact details provided above. And thank you so much again for listening to my talk.
All right. So next up is going to be Matthew Kustra. Matthew, can you please share your screen? So Matthew is a Miller Research Fellow at UC Berkeley. He's an evolutionary ecologist whose research focuses on reproductive interactions, particularly post-mating sexual selection. Take it away, Matthew, please.
Hey, I first want to thank the ASN Graduate Council for inviting me to speak at this symposium. I'm currently a postdoc at UC Berkeley, like Dylan said, but today I'll be presenting a project for my PhD that was in part supported by the ASN Student Research Award, looking at how temperature influences post-mating sexual selection in marine fish, more specifically looking at how temperature is relevant to climate change influence ovarian fluid sperm interactions. So what is ovarian fluid? So ovarian fluid is the fluid that's in the reproductive tract or surrounds the eggs. And it generally has a positive influence on sperm performance, helping fertilization success and helping sperm live longer, sperm find the eggs. And while it can help with fertilization success, it also influences the dynamics of fertilization. So say we take away the ovarian fluid, we might see that if two males mate with the same female, so one male sperm represented with orange, another male sperm represented in blue, we might see that they have equal paternity. But with ovarian fluid, that changes where the orange male is now siring more offspring. And while this is a cartoonish example, this has been documented in a wide range of taxa, from invertebrates to vertebrates, species with external or internal fertilization. In many cases, this can be adaptive, so plays a role in preventing inbreeding or as well as preventing hybridization. However, we don't really know too much about how temperature, especially in the context of climate change, might be influencing some of these dynamics. And so that's what I'm particularly interested in. And to approach this, I use the ocellated wrasse, Symphotis ocellatus. It's a marine fish with external fertilizations in the Mediterranean. And this system is good for a few reasons. First, it's relevant because the Mediterranean, unfortunately, is a climate change hotspot where it's warming about 20% faster than global average. And in the species, we also know that these ovarian fluid sperm interactions are important and post-mating sexual selection is pretty strong, which I'll talk about a little bit in a little bit. And so really it makes it a good system to ask how temperatures relevant to climate change might be influencing post-mating sexual selection. And so just to orient people, all this field work is done at a Storiso lab in Corsica, up over there. And because it's a field station, we have good data from since the 1980s. And so shown here on the left is just the temperature across within the season. So At the beginning of the reproductive season, early May, temperatures around 16 C. But then towards the end of the reproductive season, temperature increases all the way to 22 C. And so temperature is varying while they're reproducing. And so I'm really interested in how temperature might be influencing some of these reproductive patterns within a reproductive season. But as I talked about before, across reproductive seasons, we know, so since 1980 to now, temperature has been increasing, and there's been an increase in the frequency of heat waves. And so we can ask about how temperature, especially thinking in the context of climate change, might be influencing these reproductive interactions. And a little bit more about the ocellated wrasse. So in the system, we actually have different male phenotypes. So the first is a nesting male. And so the nesting male builds a nest, courts females. Females go to the nest, lay the eggs. And nesting males provide all the obligate parental care. So once females lay eggs, they go away. And so without any parental care, the nest will fail. We also have these smaller males, sneakers, and they sort of wait around for the nesting male and female to mate before jumping in and trying to release sperm to fertilize eggs. We also have this third type, a satellite male. And they pair up with the nesting male, chase away sneakers, help court females, but then also sneak mate. And so there's a lot of post-mating sexual selection, a lot of sperm competition happening in the system. A little bit more about these different male reproductive tactics. So as a juvenile, if you have fast growth, you'll be a satellite male in your first year, and then a nesting male in your second year. And these fish don't live longer than 2 years of age. If you're a juvenile male with slow growth, you'll be a sneaker male your first year. And if you survive the second year, you'll be a satellite male. So for the purpose of this experiment, I focused on nesting males and sneaker males because they represent distinct life history pathways. And we also know that this is the strongest preference difference between females have between these different male tactics. These male types also differ in sperm characteristics. So nesting males, sneakers release about 4 times as much sperm as nesting males. However, nesting males, they produce higher quality sperm, at least initially. So nesting males, they have higher initial sperm velocity. And we know how this might influence fertilization dynamics in the system. So without a varying fluid, sperm number is the most important thing. So pretty much a one-to-one correlation. So the more sperm you produce or release, the higher the paternity. And in a natural setting, nesting males will be around here because they're releasing less sperm than a sneaker. However, with the ovarian fluid, these dynamics change drastically. So sperm number stops mattering as much. And what really matters is actually that initial sperm velocity is the best predictor of paternity. And so what I'm really interested in is how temperatures, especially temperatures relevant to climate change, might be influencing these sort of interactions that we know to occur. So going back to the different temperatures that are naturally occurring, I wanted to choose test temperatures that were relevant both of what they're experiencing now as well as thinking about heat waves and future predictions. So the first test temperature I picked was 16 C, which is about at the beginning of the reproductive season, 22, which is towards the end, and 28, which represents a heat wave or a future conditions. And I want to highlight quickly that 16C, just because of the skewness of the reproductive season, most of the reproduction is happening in this part of the graph. So 16C is closest to what they would be experiencing for the most part. But at the end of this reproductive season, there's really very few active nests going on. And so to do this experiment, we took a female, collected and diluted ovarian fluid into our different test temperatures, so 16, 22, 28. And we also wanted to have a control of like just seawater. And then I took a nesting male or sneaker male, and I recorded their sperm in all different combinations. And this lets me control for individual variation. So this a single nesting male or a single sneaker, they're having their sperm measured at each one of each trial, each combinations. And I also want to highlight that the microscope is temperature controlled stage. So we're actually recording it at 16C, 22C, or 28C. And so what does this look like? So here's a video of sperm all swimming around. Luckily, I don't have to manually manually measure how fast they're moving. We have some software that will do that for us. And so it tracks the path and gives various metrics about like how progressive the sperm are, how fast they're moving. For the purpose of this talk today, I'll mostly focus on how fast they're swimming. And interestingly, I found a three-way interaction between the fluid, male type, and temperature. And so I'm going to slowly walk through what these results are. And so on the left part of the graph is going to be the nesting male. On the right is the sneaker. On the y-axis is the sperm velocity, so how fast the sperm is moving, and x-axis different temperatures. And the color is going to represent whether it's seawater or ovarian fluid. So just focusing on seawater, for both male types, sperm moves faster, independent of ovarian fluid. Sperm is moving faster at warmer temperatures for both nesting males and sneakers. If you look at ovarian fluid, for nesting males, there's actually no effective temperature anymore. So temperature does not have an influence on sperm velocity in ovarian fluid for nesting males. However, for sneakers, we see that there's still a positive impact of temperature. So sneaker males are still swimming faster in ovarian fluid at the warmer temperatures. And so we have this interaction of male tactic, ovarian fluid, and temperature occurring. But let's just focus a little bit more on that effective ovarian fluid, which we know in theory, also in this system and in most systems, that ovarian fluid should have a positive impact on sperm velocity. And indeed, we do see that at 16C, or the coldest temperature, which is closest to where they would normally be functioning, especially for nesting males. The effect size is much bigger there. However, at 22 degrees Celsius, that effect goes away for nesting males. So ovarian fluid is no longer having a positive influence. However, in sneaker males, there's still a positive influence, although it's a little bit less. And finally, at the warmest temperature, 28C, ovarian fluid is no longer having an effect. So ovarian fluid is really only having that positive impact that it should, such we know does in normal conditions, at normal conditions, and not at the warmer conditions, especially for nesting males. Now, someone interested in post-mating sexual selection, I'm really curious how nesting male and sneaker sperm differ in these conditions. And so it's a little hard to see this in this graph, so I'm going to change how I'm plotting the data a bit. So it's going to be a difference graph. So if you on this black part, you can think of it as nesting males have higher sperm velocity, so nesting males win. On the bottom, sneaker males win, so they have higher sperm velocity. And like the previous work suggests, at 16C, we do see that nesting males have faster sperm. However, at 22 degrees, sneaker males have faster sperm. And same thing with 28 degrees. And so while this is our just showing sperm velocity, we have a pretty good sense in the system how sperm velocity relates to paternity. And so I'm going to show you some posterior distributions sort of based off of some of our previous estimates on that relationship between sperm velocity and paternity. So I'm going to show you posterior distributions. So on the, you think on the left side, you can think that nesting male paternity has declined. On the right side, nesting positive numbers, nesting male paternity is increased at the warmer temperatures. And so if you go from 16 to 22C, there's about a 9% predicted difference in nesting male paternity, so a decline in nesting male paternity. 16 to 28, pretty similar patterns observing, but if you go from 22 to 28, there's no difference. And so essentially most of that negative impact of warmer temperatures is occurring at that cooler temperature of 22 degrees, right when the reproductive season would be naturally ending. And so just to summarize, looks like taking all these results into account, climate change could be undermining cryptic female choice in the system. So it's really messing up with the interactions between ovarian fluid and sperm. We're only seeing that positive impact of ovarian fluid at the coolest temperatures. Warmer temperatures is likely biasing paternity towards unpreferred sneaker males. And while this is a little bit more speculative, I'm pretty interested in understanding how this might affect population dynamics and evolutionary consequences of these interactions that I'm interested in exploring. And so I'll just briefly go over what other things that I'm currently working on. So a big part of my research program is developing mathematical models and simulations on post-mating sexual selection. specifically looking at the role of post-mating sexual selection and speciation. And currently, as a postdoc, UC Berkeley and Chris Barton's lab, I'm digging into speciation genomics and adaptive radiations, mostly focused on this Lake Chichikanab radiation in the Yucatan of cyprindon pupfish. In this radiation, there's multiple specialists, including a bivalve specialist and a zooplanktivore. And these species, this is a really young radiation, about 8,000 to 12,000 years ago. So there's been this rapid divergence and evolution. And so I'm using a combination of lab experiments as well as genomics to try to understand and gain insight into the genomic architecture, sort of the temporal stages of adaptive divergence, and try to understand the genetic origins of adaptive variation. So standing variation, de novo mutations, or adaptive integration. And I'll be presenting some of this work in the in-person portion of this conference. And with that, I'd like to acknowledge my PhD advisor, Suzanne Alonso, as well as my committee members and Team RAS's and the Team RAS. It wouldn't be possible to do this without, as well as this ASN for the Student Research Award that helped fund this project, and as well as the NSF and other my current funding from Miller Institute. And with that, I'd be happy to take any questions. And if you're curious, there's a lot more in this from this paper. It just recently got published in Functional Ecology.
All right, thank you so much, Matthew. I think we may have time for a quick questions. Do we have any question, Jacob, maybe?
Not yet, but please raise your hand or ask in the Q&A if you do.
Sure. In the meantime, our next speaker is going to be Nitin Ravikanthachari. So if you can please share your screen, that'd be helpful. Okay, Nitin is currently a postdoctoral research associate at the University of Montana. In his research, he investigates how environmental changes influence biotic interactions that shape Lepidoptera adaptation and evolution. Take it away, Nitin, please.
All right, yeah, thank you. Hey everyone, I'm Nitin and I'm a postdoc at University of Montana. Today I'll talk about how genetic and environmental factors shape species interactions at different biological scales. Butterflies and the host plants that they feed on are a classic example of coevolution. And for my PhD, I worked on cabbage white butterflies, specifically a species known as Pyrus macdonui. And this is a butterfly that occurs in the Rocky Mountains in the US. And these butterflies are Brassicaceae specialists. That is, they are specialized on mustard family plants. And they feed on the mustard plants using nitrile specifier proteins, which they use to detoxify the glucosinolates, that is, mustard oils, that's present in these plants. The females of this species oviposit and the larvae feed on a range of native mustard plants, and they use the same mustard oils or glucosinolates as cues to find these plants. And I was interested in a specific mustard known as Thlaspi arvense that this butterfly interacted with. And this plant is native to temperate Eurasia and was introduced into the US in the late 1800s. And this plant contains synegrin as the main glucosinolate. So what was really cool about this interaction? The females of this butterfly recognize Thlaspi as a host plant because it shares a lot of Q similarity with native plants. And the Q is most likely synegrin, the molecule or the glucosinolate that's present in both these plants. Most of the larvae die before pupation when they feed on this plant, and thus it leads to an evolutionary trap. However, we know that larvae vary in the time that they take to die when they're feeding on this plant, and they can be rescued if they're provided with native host plants. So I was particularly interested in understanding what the responses of the larvae were while feeding on this plant, and what the responses of the females were when they were laying eggs on this invasive plant. So to do this, we conducted a simultaneous oviposition choice experiment with the females where we gave them both the plants and asked them to lay eggs. We let females lay eggs and at least 50 eggs minimum. And once they laid that, we removed the females and we quantified preference based on how many eggs were laid on each plant. And we did RNA sequencing on the antennae and the forelegs specifically to look at sensory gene expression. Additionally, we also conducted a no-choice larval performance trial where we gave third instar larvae with either of these two plants. We let them feed for 24 hours, and we did RNA-seq on the mid-cut after 24 hours. So this is what we found. We found that several genes were differentially expressed in the larvae that were feeding on these two different plants. But however, we did not find any differential gene expression related to female preference on either of these two plants. So that was very surprising for us. Coming back to the genes that were differentially expressed in the larvae, they could be grouped into 3 different clusters. And additionally, the expression pattern clustered based on the treatment, indicating that the larvae that fed on a particular plant had similar responses. However, we did see some variation in those larvae that were feeding on the invasive plant. So of the 3 clusters, the genes in the first cluster were enriched in larvae feeding on the native plant and were mainly related to expression of specialized phase 2 detoxification enzymes. And these enzymes are critical in specialist herbivores to help them detoxify the chemical compound that they have evolved to feed on. Conversely, the 2 remaining clusters were enriched in larvae feeding on the invasive plant, and these genes were related to phase 1 and phase 3 detoxification enzymes. And phase 1 enzymes are highly expressed in generalist herbivores, and phase 3 enzymes are mainly involved in excretion. So the larvae that were feeding on this mustard plant were behaving like a generalist, and they were struggling to feed on this plant. Since we did not see any differential gene expression in the female preference, we went ahead and looked at allelic differences to test if it was the allelic differences between female preference that was driving their choice. So to do that, we used the same gene expression data set and we called SNPs. And we looked at an FST plot looking at differentiation, where basically the plot here shows genetic differentiation between females with different preference. What we see is that there were multiple SNPs that were differentiated between females with different host plant preference. And a lot of them were actually present on the sex chromosome. When we looked at what these genes were or where these genes were enriched, we found that a lot of the genes were related to signal transduction. So this suggests that it was allelic differences that was driving female preference rather than differential gene expression. So for this part, what I would like to say is that the decoupling of oviposition preference and the larval performance is the main reason for this evolutionary trap. And based on whatever I've shown you, I think the likely escape from this trap will involve larval ability to feed on the invasive plant and or selection against the sensory alleles in females preferring to oviposit on this invasive plant. So there's been a lot of theory and research which says that error-prone oviposition is the fuel for evolution of host plant range, and I think we have provided a mechanic understanding as to how error-prone oviposition can actually help expand oviposition range in herbivorous insects. What I haven't told you so far is that Pyrus butterflies, along with almost 50% of all known arthropods, are associated with an endosymbiont called Wolbachia, a bacteria that lives inside the cells of their host. And this is what I'm continuing my work with during my PhD, during my postdoc. And Wolbachia are maternally transmitted, and they are the most widespread endosymbiont in nature. The main reason for their success is that they have evolved a range of strategies to influence host reproduction that enhance their own transmission through host populations. A key feature of Wolbachia is that most of these associations are facultative and the host does not really require them to survive. So I was interested to see if frequencies varies across different insect orders. So here is a phylogeny of all the major insect / arthropod orders, and on the y-axis is population mean Wolbachia frequency, and the error bars represent the maximum and minimum Wolbachia frequencies observed. And as you see, most species have Wolbachia in which the Wolbachia varies significantly. So this, brings up to the question as to what contributes to this frequency variation. So while we know many factors can influence Wolbachia frequency, we know that maternal transmission, that is how reliably Wolbachia is passed on from moms to offspring, can vary widely and is influenced by environmental factors. So Ari Hoffman and his lab have been looking at Wolbachia frequency variation in a specific Wolbachia that occurs in Drosophila melanogaster, and they have been tracking this since 1988. And what they found was that Wolbachia frequencies are higher in tropical regions and they decrease sharply in temperate regions. So a previous postdoc in the lab measured maternal transmission, shown here on the y-axis, at different temperatures. And what he found was that maternal transmission is highest at high temperatures, and then it decreases sharply at low temperatures. So expanding on this, I wanted to test if frequency, Wolbachia frequency also varied across all continents. So to do that, what I did was I expanded this data set to look at Wolbachia frequency populations in melanogaster across 248 locations spanning 42 years. And this includes previously published reports, PCR screening of multiple new populations, and genomic screening of whatever was published online at NCBI. And I specifically tested if latitude varied with Wolbachia frequency across continents. And the x-axis here represents absolute frequency, and the y-axis here represents, absolute latitude, and the y-axis represents Wolbachia frequency. And what we find is that there is a clear support for non-linear latitudinal climb in Eastern Australia, but there is no influence of latitude on Wolbachia frequency in any other continent. Since latitude represents a coarse proxy of various environmental factors, we then tested if specific environmental factors influence Wolbachia frequency. So to do that, I used the 19 bioclimatic factors as my variables, as they represent long-term climates experienced by host populations. Of the 19 variables, we found that frequency was influenced by 3 factors, which we think act through influencing host biology instead of acting on Wolbachia directly. So here, the x-axis is z-scored. So negative values indicate lower than average values, and positive values indicate higher than average values. And the y-axis is predicted Wolbachia frequency. First, we found that precipitation seasonality was positively associated with Wolbachia frequency. So what this means is that regions with distinct rainy and dry seasons have higher Wolbachia frequency, and this is because seasonal abundance of food, is a critical thing for hosts, and which can drive host population size and in turn lead to higher transmission and frequencies. Second, we found that precipitation during the driest quarter was also influencing Wolbachia frequency positively, and precipitation during the driest months provides resources and prevents starvation, which can increase host activity and in turn maternal transmission and Wolbachia frequency increase. And finally, we found that mean temperature of the wettest quarter was associated with Wolbachia frequency, but only in Australia and not any other continent. And this is interesting because higher Bio8 values, or mean temperature of the wettest quarter, in Australia occurs during the warmest months, which coincide with peak host activity. Whereas in other continents, it is decoupled because it can occur in any other season, such as winter or spring. So it is not directly testing host activity. So moving on, I next wanted to test if Wolbachia genomic variation is influenced by environment, similarly to what we see with Wolbachia frequency variation. So to do that, I called SNPs and looked at, I conducted environment allele associations using latitude and bioclimatic factors as the variables. So of all the factors, we found about 38 SNPs that were associated with latitude. And all of these 38 SNPs had higher derived allele frequencies at temperate latitudes and lower derived allele frequencies at tropical latitudes. However, this association disappeared when we corrected for lineage structure. What I mean by lineage structure is illustrated here. So here is a phylogeny that illustrates the major recognized clades of Wolbachia or WML cytoplasmic lineages. All of our environmental association traced to this particular lineage in Southwest Europe. And it is interesting because the variant called WMLCS was rapidly replaced by the current variant about 8,000 years ago. And this is basically a remnant of that particular variant. So this reflects an incomplete global sweep rather than an independent environment allele association. So with this, what I'm shown is that latitudinal cline that we see in a lot of Wolbachia can be decomposed to specific environmental factors, and precipitation regimes shape global Wolbachia frequencies through influencing host nutrition. And the latitudinal climb we observe in Australia is because of Australia-specific vent temperature season. And then the genomic variation that we see is because of cytoplasmic sweep rather than independent environment associations. And with this, I'd like to thank a lot of people, both my PhD and postdoc, who have helped me with this work. And I'm happy to take any questions if there's any time left. Thanks.
Sure, we may have time for one quick question. Thank you, Nitin. And next up is Ratna Karachi. Ratna is currently a postdoctoral researcher at the University of Helsinki. Her work is focused on sexual selection, intrasexual competition in both males and females, and mate choice. So Ratna, please take it away.
Hi, my name is Ratna Karadki, and I'm a postdoc, as Dylan said, at the University of Helsinki. But today I'll be talking to you about my PhD research, which I did at the University of Illinois with Dr. Becky Fuller, where I looked at the aquatic lighting environment and its effect and depth, and their effect on the influence of nuptial fin coloration through phenotypic plasticity. A common phenomena that's seen across organisms is the presence of stable trait variation, both within species and within populations of species. And there are two broad mechanisms that contribute to this kind of trait variation. So you see genetic polymorphisms, where the genotype of an individual largely determines the phenotype, as seen in these Dalmatian wall lizards and their skin coloration. The other two examples shown here are examples of polyphenisms or phenotypic plasticity, where individuals of the same genotype can have different phenotypes depending on the environment, and this variation in the phenotypes can be discrete. So in the Daphne example, we see that the presence of predator cues makes them develop these altered head morphologies, and in the monkey flower, changes in the photoperiod dramatically alters the shape and size of the flower. So this kind of persistent trait variation raises a central question in evolutionary biology, which is what sort of mechanisms maintain this trait variation over time? And specifically, how does the environment contribute to this kind of variation? And while these examples shown here are striking and have been informative, there is another very compelling system which I study, which offers a powerful lens to address these questions. This is the bluefin killifish. As you can see, there's a remarkable variation in fin coloration. These are all males of the species, and the interesting thing is that they show polymorphism in the fin color of their anal fin, which is a large fin below the belly. So we see red, yellow, and blue male morphs. In the wild, bluefin killifish inhabit rivers where there is a lot of variation in the lighting environment. So they are found in crystal clear spring rivers, tannin-stained swamps where the water is brown but not turbid, And in some places, there is a confluence of spring and swamp rivers as well. Red and yellow males have been found across all rivers of different lighting environments. However, the abundance of the blue male morphs is associated with the lighting environment, where as a river becomes less tannin stained or the abundance of UV and blue wavelength light increases, the percentage of blue males decreases in those populations. So each circle here is a population that has been censused and the percentage of blue males there. There are two axes of variation that contribute to this color pattern that we see in the species. The red versus yellow phenotype is genetic, with the yellow being dominant to red. And then you have the blue versus non-blue phenotype, which is plastic. And we know this from a previous breeding experiment that my advisor did, where we brought fish from the wild and reared them in the lab under different conditions. Before going forward, I just want to mention that you'll see a lot of tea-stained lighting environment on the x-axis in my graphs, and that's because we actually do use unsweetened decaffeinated iced tea powder to mimic the swamp-like tannin-stained environments in the lab. It's a really cheap and easy way to mimic their natural lighting environment. So when we bring fish in from the wild and have males of different colors breed and produce offspring which are reared under different conditions, we see that those offspring that were reared under the tea-stained conditions are much more likely to express blue anal fin coloration. We also see that there is some amount of variation as well, even within a population. Additionally, we've observed behavioral correlations of these different fin colors specific to lighting environments. So if you look at this graph, in blue coloration are situations where a blue male outcompetes non-blue males when it comes to competitive interactions, which are crucial for them to acquire territories. And when one of their predator fish, which is bass, was presented with males of different colors under different conditions, it's seen that these predators are less able to perceive blue males under the tea-stained conditions which mimic the swamps. So this indicates that this blue phenotype and potentially plasticity in blue phenotype is adaptive. Yet we see a lot of variation here in this plasticity. So the overarching question of my research was to understand how variation in different environmental variables can potentially promote or limit the expression of these plastic and color polymorphic traits. So compared to terrestrial lighting environments, aquatic lighting environments are very variable. And one unique factor is the presence of dissolved substances. In lakes, we have dissolved algae. In swamps, we have tannins due to the presence of decaying plant and animal material. So not only does this lead to a difference in the overall coloration between the different kinds of rivers and habitats, we also see microhabitat variation due to the interaction of depth and the presence of these dissolved substances. So on this graph, on the y-axis, we have downwelling irradiance, which is essentially measuring the abundance of different wavelengths of light, which is represented on the x-axis. And the different colored lines here represent depth in the water column. So at the very top of the water column, which is this lightest line here, the different wavelengths are pretty much similar, the lighting environments between the two conditions. But if we look at just 38 centimeters deep, which is not even half a meter deep in the water column, we see that short wavelength light is significantly reduced in the swamp conditions, but not so much in the spring. So in this study, I examined the relative contributions of dissolved organic substances, depth, and the interaction between these two variables on plasticity and blue coloration. I also wanted to quantify how these factors influence variation in plasticity within this population across different families. So to do this, we collected blue male morphs and females from the Everglades, which is a swamp population, set up 15 crosses, 13 of which actually ended up spawning in 4 treatments that we had here. So we had 2 lighting environment conditions, which is clear and T, and we had 2 depth conditions, top and bottom of the water column, and we manipulated the depth by altering the depth essentially at which we presented the spawning substrate to the fish. We reared all the offspring until adulthood and noted the male coloration. So here you can see the eggs on the spawning substrate. You can see freshly hatched fish, which are just big eyes and a little tail. And we raise them until the adult males express color on their anal fins. euthanize them, and record the coloration of these male offspring. So let me just set you up with some of the predictions that we had going into this study. We know that the abundance of short wavelength light decreases as we go from the clear top condition to the T-stain bottom condition. When it comes to the macro habitat level effect due to the presence of tannins, we know from earlier studies that for the proportion of blue offspring, there should be more in the T-stain conditions. We expect that there would be more there compared to the clear conditions. Now, when we add in the effect of depth, we expected that the proportion of blue suns would be inverse to the abundance of the short wavelength or blue light. Okay, so moving on to the results. The first pattern that we see is that there are more blue suns in the T-stain lighting environment, which is a swamp-like condition. So here on the x-axis, you have the 4 treatment conditions, and then the blue coloration represents the presence of blue suns within a family. So we see that overall there are more blue suns in the T-stain condition compared to clear. Surprisingly, we did not see an effect of depth within the T-stain conditions. However, we saw a big effect of depth in the clear lighting conditions where there were no blue suns at all in the clear top treatment. So this graph is showing you the proportion of blue suns of each family across the different conditions. Each line here indicates a family. So you can see that there is substantial variation in plasticity across families, even within this highly plastic population. And this is just separating all these families out. So we had some families where they had no blue suns at all across the board. This is despite the fact that all these crosses had blue dads. And then we saw different extents of plasticity across some of the other families. And some of the more plastic families tended to produce more blue suns at the top of the water column in the T-stain lighting environments, although this effect was not significant. So if we go back to our prediction schematic, what we actually see is that there is this big macro habitat difference due to the presence of tannin, where in the tannin stained conditions, we have significantly more blue suns compared to the clear. We don't see an effect of depth within the T-stain condition, but we do see this effect of depth within the clear conditions, with this clear top treatment having no blue suns. So it seems almost as though there is this upper limit or a threshold for the upper limit of abundance of UV and short wavelength blue light, beyond which the production of blue suns seems to be inhibited. An interesting aspect of this study design. So the fish were held at the depth treatments for mostly a very early part of their development. So the eggs were spawned on the spawning substrate, which were kept at different depths in the water column. And soon after hatching, when the fish couldn't swim too much. So it's only within this short window during their egg stage and soon after hatching that they were at the depth treatments, after which they were free to swim about the entire tank. Despite that, we saw the effect of depth in the clear condition, indicating that even this very early stage can be very important for expressing the plastic phenotype and potentially preventing the expression in the clear condition. And very quickly, I'll just mention another study where I was looking at some potential mechanisms underlying this fin color variation. And looking at fin tissue transcriptomes, we saw that in blue males, there were a couple of genes that were significantly upregulated compared to non-blue males. The first is a key transcription factor gene, which not only affects downstream deposition and production of the cells that make the structural blue coloration, but also influences other transcription factors involved in this process. And we also saw that an extraocular opsin gene, so these are involved in non-image forming light perception. This extraocular opsin gene RGRB was significantly upregulated in blue males compared to non-blue males, indicating some potential mechanisms in which they could potentially perceive their environment and modulate this phenotype. So to summarize, through the breeding experiment, we saw that macrohabitat differences were the primary driver of plasticity in fin coloration. However, we do see the effects of the interaction of depth and lighting environment, specifically in the clear condition. And again, we documented tremendous genetic variation in plasticity within this swarm population. And we don't see any blue suns in the treatment with the most amount of short wavelength light. And we also have some indication from this study that the critical window for the induction of plasticity can begin fairly early on in development. Yeah, and I'd like to thank everyone who was involved in this study. I'd like to thank my advisor, Dr. Becky Fuller, the Fuller Lab, all the lab members, all the undergrads who helped raise thousands of babies and clean hundreds of tanks, and of course ASN for funding this research and giving me the opportunity to present this talk. And with that, I'm happy to take any questions if there's time.
Thank you so much, Ratna. That was very interesting. Yeah, we have time for questions. Do we have any question?
We
Jacob?
don't have any yet, but yeah, please raise your hand or throw it in the Q&A.
Sure. Yeah, the next speaker is going to be Taylor Zallek. Taylor, you can share your screen right now, and then we'll start at 6:30.
Good. Can you hear me, Dylan?
I can, yes.
Perfect. Yeah, just give me the thumbs up when.
Will do. Yeah. Well, Taylor Zallek is a postdoctoral researcher at the University of Minnesota. He's an evolutionary ecologist whose research focuses on how rapid evolutionary processes shape community and ecosystem dynamics. Please take it away, Taylor.
Great. Thank you. And thank you for the invite. I really appreciate the opportunity to be able to speak to you virtually. Really appreciate the funding from ASN, and it's great to kind of give back in a very small way. But yeah, I'm currently a postdoc at the University of Minnesota, but I'm going to be talking to you about the research that I conducted as a PhD student at the University of Pittsburgh, where I worked in Dr. Martin Turcotte's lab. As a researcher, I'm an evolutionary ecologist who's particularly interested in the ways that eco-evo dynamics can interact to influence kind of biological invasion processes. So I'm going to be talking about invasibility. It's not quite a very household term. You might have heard of invasiveness. But invasibility is the susceptibility or kind of resistance of communities to establishment of introduced populations. So you might imagine that communities with different assemblages might be more or less resistant to invaders. And we can measure these differences and track them and statistically quantify them. And it can be important for a lot of reasons. You know, these are from the very applied. It allows us to better predict invasions, conserve native populations, improve kind of restoration efforts, but it also has a lot of kind of basic science components to it. It allows us to kind of study competition, coexistence, community assembly, priority effects. All of these things are at play here. And I'm kind of adding sort of the eco-evo dynamics to this as well. And we know that invasibility can be altered by ecological interactions in diverse communities. There's many examples of this, from altered plant pollinator networks that reshape invasibility by non-native weeds, excuse me, or through indirect trophic cascades where small islands invaded by ants can have massive impacts on the introduction of invasive snails and other things. So we know that invasibility can be shaped by ecological interactions. What is less known is whether or not it can be shaped by evolutionary interactions, in part because it's really hard to measure. There's some evidence from observational studies that hints that this could be really important. So for instance, populations of native plants that are exposed to invaded plants might evolve to increase their tolerance or competitiveness to those invaders. But these are just single species, single populations or isolated populations, and I'm really interested in kind of this broader kind of community component or community metric that is invasibility. And I want to take a slight detour because I think it's appropriate for this audience. So what motivated a lot of this research was actually a side conversation I had at the virtual Asilomar in 2020 when Kelsey Gleiberger, who's now faculty at Arizona State, she was a PhD student at the time, and she reached out to me and said, "Hey, your talk made me think about this study that I read. It's from 2014 that measured how the impacts of genetic diversity in invading populations and how that mattered for invasive success." And she was really like, what if we did the opposite and think about the residents? What if we measured how residents perform and resist invaders based on the genetic diversity that's present? And so we performed this meta-analysis that looked at 37 studies with 109 effect sizes, and we predicted that higher genetic diversity in resident populations would probably decrease the likelihood that invaders succeed. What we found was different. It wasn't what we expected. So what I'm about to show you here is these sort of differences in the negative effects that increasing genetic diversity can have across taxa, across study system, et cetera. And I'll just summarize it quickly and say that increasing genetic diversity had little to no effect, a slight negative effect, but it really didn't matter. And that kind of caused us to scratch our heads and go, what could be going on here? This kind of, not only did it violate our hypotheses, it violated our theory. And what it got me to think more about was maybe just manipulating the genetic diversity of single resident populations isn't enough to tell the full story, that there's other ecological components. So thinking about how does genetic and species diversity, how do they interact to influence invasibility? And what about evolution, the genetic change through time? Maybe that's really important too. And so this is where complexity, I think, is important to include in our experiments. To do this, I needed to use a study system that was amenable. So I used duckweed. Shout out to Takuji for the great introduction of this system. And also shout out to ASN for funding 2 duckweed proposals. I really appreciate that as well. So I used two native duckweed species in North America, commonly found together. These are Spirodella polyrhiza and lemna minor. They reproduce vegetatively very quickly, short generation times, and we can actually establish populations with known genetic variants, allow them to evolve over time and track that evolutionary change in a highly replicated fashion. We can do these experiments in these reconstructed we do them in cattle tanks. This is at the Pymantuning Lab of Ecology in Northwest Pennsylvania, belongs to the University of Pittsburgh. Another big funder of my research. And so we can actually bring that noise of and the variation from nature into our studies, because we think that's really important too. It's not just invaders that native species are evolving to. They're also evolving to different, changes in heat and rainfall and all kinds of abiotic factors too. See if I can get past. There we go. But to measure invasibility, we also need invaders. And so I adopted these non-native species, floating aquatic ferns called Salvinia. Salvinia float on the water surface. They reproduce vegetatively very quickly. They have a very similar niche as duckweed and kind of compete for the same space. And and it's also important to note that these are really problematic species, particularly in the southern United States, where they clog waterways, they outcompete native plants. They're a big problem, but they're not in Western Pennsylvania, where we collected our native duckweed genotypes from. So important to note that the genotypes that we used in our study had to our knowledge, no evolutionary or overlapping history with these invaders. So to address this first question, how do genetic and species diversity interact to influence invasibility? The first thing I had to do is I had to find a really rock star research intern. This was an undergraduate by the name of Ashlyn Moretti. She's now graduated and doing cool stuff of her own. But we were able to conduct an experiment where we varied components of the resident populations and communities. So if the yellow represents invaders, the color surrounding them represents the residents. So not only did we alter the species identity of the native populations, but also the species diversity. And then we factorially altered the genetic diversity of these populations and communities as well. And what we predicted or hypothesized was that the most diverse, both genetically and species-rich communities would be the best at resisting invaders. They'd be the least invasible. So that was the prediction. And so how we measured that is we measured as a proxy, we measured change in invader biomass over time in these communities and populations. And what we saw initially is, at least for our single genotype populations, we see a ton of variation. But this variation, then we begin to see these negative slopes. So as in our single populations of both Lemna minor and Spirodella polyrhiza, we see a decrease in invasibility with increasing genetic diversity. That's what we predicted. And we also see that there's also different intercepts. So we see that Spirodella polyrhiza is slightly better at resisting these invaders than Lemna minor. But when we add two native species to this mix and we increase the genetic diversity, we see the opposite occur. We get this interaction, such that the most diverse native populations and communities are among the most invasible. And this was not expected, but I think it just goes to show that these higher order interactions are important and hard to predict. And competition among native species should be considered when we're thinking about processes like invasibility. And yeah, so kind of unexpected. But now, if we're thinking about eco-evo interactions, what about not just the genetic diversity, but the genetic diversity change over time? How might that influence invasibility? So to do this, we did some experimental evolution with native populations and communities exposed to two Salvinia species for a period of 7 weeks. We used 7 genotypes of each species. We then introduced, some invaders. We allowed them to evolve. We babied them and took care of these communities. This was conducted at PLE as well. In these buckets, we tried to minimize a little bit of the radiation with some shade cloths. We tracked them. And the first thing we wanted to do was identify evidence of rapid evolution. So to do this, we measured changes in genotype proportion over that 7-week period from their initial genetic proportion. So among populations of Lemna minor that evolved with Salvinia minima or molesta, we see, I'm just going to summarize this very quickly. It's kind of hard to read this fast. We see a significant effect of genotype, but no interaction with evolution treatment, suggesting that some genotypes evolved or were selected for, some were selected against, but it was consistent across our evolution treatments. So they evolved, but they evolved in similar ways. Now, for Spirodella polyrhizae populations, the story was a little different. We see this significant interaction. So not only did your genotype matter, but it also mattered who you evolved with, which invader you evolved with. So the genetic change was much stronger here. After conducting our experimental evolution, we common gardened these populations, not just populations separately for few generations. We had to remove the invaders. We separated the residents from each other. This was really tedious. And then we recreated these communities, reestablished them, and invaded them in a factorial design that allowed us to invade with Salvinia minima, molesta, a co-invasion with a little bit of each. And we also, in order to test evolution's effect, we recreated those initial genetic compositions, kind of the ancestral compositions, to have a non-evolved treatment as well, and invaded them. And this factorial design allowed us to ask questions like, does evolutionary history, focal invader, or their interaction best determine the invasibility? And so we allowed these to run for another 2 weeks, and then deconstructed these communities and measured and changed in invader biomass over time. And I'll cut to the chase because I'm running up on time. But what we found was that the only factor that mattered was the evolution treatment, regardless of who you were invaded by. Evolution history was the best significant predictor. If you evolved with Salvinia minima, you were generally just more resistant, less invasible. And we also zoomed in on the kind of co-invasion treatment, dissected what could be going on here. I'll go through this very quick. What we found is that the Salvinia minima that we invaded with, not so different, but the Salvinia molesta here really tells the story. So those that evolved with Salvinia minima are just generally less invasible, even to things they didn't evolve with. Takeaways: genetic and ecological interactions can dictate invasibility in non-additive ways. Evolutionary history matters for determining invasibility. But one quick plug, the support from this research grant also supported a number of students that I was able to hire and train. And so this, and I, gaining mentorship experience. And so I just wanted to go on record as saying that, these grants fund a lot more than just the students that are receiving them.
Thank you so much, Taylor. That was a great talk. We do have time for questions. Please feel free to use the Q&A chat box for questions. Do you see any question, Jacob, by any chance?
No,
I
not
don't.
yet.
Okay.
Not yet. Well, I'll just say one more time, thanks everybody for attending late on a Friday or from wherever you are. Maybe it's early
Yeah,
now.
thank you, Taylor, for
Take it easy.
making the effort. That was great. And closing up this fascinating symposium, we have Julia Harenčár. She's a postdoctoral fellow at UC Davis, and she's an evolutionary biologist passionate about combining ecology with genomics to understand plant evolution. So, Julia, please take it away.
Thank you very much for that introduction, and I'm excited to be here today. I am indeed currently a postdoc at UC Davis, but like a lot of you, I'll be sharing work that I did as a PhD student at UC Santa Cruz on polygenic divergence, enabling species differentiation despite gene flow in these really cool tropical plants. I like to start talks with acknowledgements just because we all know this work doesn't happen in a vacuum, and certainly my work would not be possible with all the people and funding sources on this slide. And of course, a big shout out to the Student Research Award. And I'm very grateful to the Grad Council for inviting me to be here today and share the results of that award with y'all. So when thinking about and studying evolution, we're seeking to understand how new species form and how they are maintained as distinct. And there's increasingly abundant evidence that prezygotic barriers tend to arise before postzygotic, such that you can still get gene flow between early diverging species. And these prezygotic barriers often involve trade-offs where there's adaptation to one environment that precludes adaptation to another, which can lead to ecogeographic isolation, which is simply geographic isolation due to differences in habitat. So you might imagine a pink cartoony habitat and a yellow habitat. And there's some group of organisms adapted to that pink, another to the yellow. And because of trade-offs, should an individual move from one into the other, it's unlikely to be able to survive. And this sort of ecogeographic isolation is very common. We see it in, for example, serpentine and non-serpentine plants, benthic and lemnitic fish like sticklebacks. And in those examples and many others, those two habitats actually occur quite close to one another and within dispersal distance of the two species, which opens the door for the potential gene flow between those two species, particularly when they lack these prezygotic barriers. And it turns out this sort of gene flow between species, also called introgression, is really common across the tree of life. There's all sorts of examples now ranging from, human lineages to plants, salamanders, insects, etc. And it begs the question, how are these species remaining distinct when they're experiencing this introgression? And the majority of work has focused on systems where reproductive isolation is driven by a few very large effect loci, and they see these islands of divergence at those particular loci. But there are many systems where divergence is actually really polygenic and controlled by things like divergent habitat adaptation. Certainly, this is true in the tropics, where we have lots of close relatives coexisting, but partitioning habitat somewhat differently, or in the case of costus, these beautiful spiral gingers, maybe having different pollination syndromes, et cetera, and there are potentially many genes underlying that isolation. I study specifically these two species of costus, Phyllocysimus and Alenii, and I'll try and make that the last time I say those long scientific names, and I'll call Alenii the pink species and Phyllocysimus the yellow. The pink species is adapted to perennially wet forest understory, and that sort of isolates it from Phyllocysimus from the yellow species, despite sharing a range. Alenii's range is the pink species range is entirely within the yellows. And the yellow, conversely, grows on seasonally dry forest edges. So think yellow dry. Previous work that I got to build off of by Dr. Grace Chen did big reciprocal transplants of these two species and found that Phyllocysimus, when growing in Alenii habitat, can't survive, and vice versa. Phyllocysimus habitat is not a good place for Alenii. And this strong habitat isolation occurs, like in those systems I mentioned, at sometimes a relatively small spatial scale. And I say relative to the pollinators that they share, which are these orchid bees that can travel quite far distances. And they don't have any prezygotic barriers. They make very happy hybrid individuals, some of which had been noted in the wild by Grace Chen. So I picked this work up starting with just basic characterization of what the extent of introgression is between the two species, and also whether it's only recent or also ancient. And then asking questions about how adaptation and the genetic basis of adaptation interfaces with selection to create patterns of introgression. So a quick introduction specifically to the plants that I collected. I got samples from Alenii from the pink species from the north side of the Panamanian Isthmus, where there is the influence of Caribbean storms giving it perennial rain, and the yellow species tends to grow on the southern side of the isthmus, where there's seasonal drought. And right in the middle of that gradient of precipitation were these putative hybrids that I collected. And I'm going to just cut to the chase here because I have quite a few results I want to share with y'all. They are indeed hybrids. And this is a molecular PCA. The x-axis here is just showing that the majority of the variance between these individuals is described by their species identity. Alenii on one side, yellow species on the other. But most of these hybrids are right near the center of that axis, indicating that they could be just recent hybrids, early generation. And to evaluate that, I used an ancestry HMM hidden Markov model. Happy to talk about that if people are curious, but I won't go into details. I'll just show you some examples. This is just going to be one chromosome, and the x-axis here is position along that chromosome, and the y-axis only has 3 places we care about. Homozygous, their Phyllocysimus ancestry will be 1, heterozygous will be 0.5, and homozygous for Alenii will be at the bottom. And what we see is that most of the individuals, 5 of them, these hybrids, look like this. So that is very clearly an F1. They're heterozygous across their whole genome. Mom was one species, dad was the other. We see 2 back crosses to Phyllocysimus. So, one parent was a F1 hybrid, the other Phyllocysimus. And then some 4 of these further generation hybrids, but always with these very large ancestry blocks, indicating again that these are indeed recent hybrids. So I wondered, could this simply be because of the history of the land use history of the area where I'm collecting? The Pipeline Road was built in World War II as an alternative option for moving oil across the isthmus in case the canal got bombed. And today it provides a sort of unusual light gap that's maintained as sort of a highway through this precipitation seasonality gradient. So I took all of the clear hybrids and removed them entirely from the rest of the analyses you'll see. I also removed some sort of questionable individuals from either species and used an ABBA-BABA analysis, which I won't explain in detail, but essentially uses the phylogeny and an in-group to evaluate how likely hybridization is, gene flow is between your focal species relative to patterns of incomplete lineage sorting, and found that absolutely there's very clear hybridization between, or introgression historically between these two species, which makes a lot of sense given this heavy overlap of the two ranges and the large distances that the pollinators can move. I then wanted to break that down more granularly across the genome. So I used a metric for looking at introgression that is windowed across the genome. And all those red points are basically representing sections of the genome that show elevated introgression. And you can see it's pretty widely distributed across the genome. There's maybe a couple chromosomes with less, but by and large, we see it everywhere. And this is sort of mirrored in some ways by divergence. So both average nucleotide divergence difference in DXY and pop differentiation in FST don't show these clear islands of divergence that other systems have seen as sort of these critical points in the genome that maintain differentiation. And so to connect these patterns with ecogeographic isolation, I did a huge admixture mapping experiment, mapping the genetic basis of a number of key adaptations to these different habitats, largely focused on ways in which the yellow species handles drought, but also some thickness and toughness, which is associated with differences in the herbivore environment, which is more intense in the wet habitat. I won't go into a bunch of detail about these traits. If you're curious, I have lots of data and reasons behind each one of them. But for now, I'll just say these are important for habitat adaptation and jump again to a results slide. If we could start by focusing on this plot on the left, I actually meant to hide this one for now, and look at these open circles versus the closed circles. The closed circles are showing the amount of phenotypic variance explained by the QTL, and the open circles are showing the unexplained variance. And you can see that those open circles by far are the highest, right, compared to the QTL. And because that unexplained variance could be environmental variability or phenotypic plasticity or polygenic effects. And I did characterize as much as possible environment within this greenhouse experiment where I tracked cohorts of plants that were experiencing the same conditions and was able to include that as a variable, meaning that most of that unexplained variance is likely polygenic effects on these different traits, which also fits the literature for some of these more complex traits. They tend to be polygenic. Further, these scans here are showing first the QTL peaks that are above our threshold that get included in the analysis, but also pointing out that there are some, sort of close sub-threshold peaks and also various medium peaks across the genome. It's not like we see one or two clear peaks and nothing else going on. And that again sort of fits what you would expect to see if there are lots of different, either additive contributing loci or some epistatic loci. And when we take these QTL regions that we have, so this sort of some portion of the diversity in the trait is explained by these, and we put it over patterns of differentiation across the genome, we don't see that every QTL interval has a clear peak of divergence. Occasionally, there is elevated FST within those windows, as you can see here, but it's not more than what you might expect by chance if you just plopped windows of those size down across the whole genome randomly. To look at that another way, we can see that this is a distribution of the FST in 1 centimorgan windows across the whole genome. So basically saying, the average FST across the genome is around 0.3. There's some windows higher and lower, but our QTL intervals, credible intervals, have averages right around the average across the genome. So they're not showing this clear pattern of elevated divergence that you maybe have seen in other systems. And I really think this makes a lot of sense and is probably fairly common because of the components that make this system so polygenic and that make selection on their habitats somewhat variable. So one factor here is repeated introgression. These have been hybridizing in different places, different points in their range under slightly different conditions. And then there's the potential for gene flow between those different hybridization events. Both theory and empirical work has shown that repeated hybridization events, even with the same conditions, can result in different patterns. Further, adaptation to the same environmental conditions in different populations can have different polygenic bases. So if you consider those things together and mix them up, you end up with a sort of more messy and less clear selection on any one locus. And yet, the species do remain really clearly isolated by divergent habitat adaptation. And so we think what's happening is that polygenic divergence is creating a diffuse pattern of selection across the genome where there's sort of widespread selection against introgression, but at any particular locus, it's not very strong, and the potential for adaptive introgression enables some gene flow in those regions, or even just neutral introgression when it's not near a sort of critical mass of these selected loci. And with that, I have no idea how time was because I forgot to restart my timer. But I'll take questions, or if I may, I'll leave this up and say that what I'm currently working on is totally different and is using herbarium specimens to look at hybrid evolution in wild radishes. So I'm building genomic time series, watching how basically the opposite pattern arose, where instead of clear differentiation being maintained, we have the entire loss of a weedy species and a total hybrid takeover. And I'm expecting that to look different under very dramatically different environmental conditions in these different parts of California. Okay, thank you.
Yeah, that was great. Thank you so much, Julia. And we do have questions for Julia. We have 2 minutes, so please feel free to ask your questions using the Q&A chat box. And that was an amazing set of talks. On behalf of the American Society of Naturalists, I can tell you that we are so happy to see you being so successful throughout your careers. If you guys are going to go to the meeting in Cleveland, please feel free to send me an email. It'll be so nice to chat with all of you at the meeting and in person. So we still have time just in case people have questions. All right. I don't think we have questions. So thank you so much all for attending, and you'll have a good weekend.